Good afternoon, and welcome to the Department of Medicine Grand Rounds. Please take a moment to silence your pagers and mobile devices. Today's Institute for Clinical and Translational Research in Medicine Grand Rounds will be presented by Dr. Christopher Chute. Dr. Chute is the Bloomberg Distinguished Professor of Health Informatics, Professor of Medicine, Public Health, and Nursing at Johns Hopkins University, and Chief Research Information Officer for Johns Hopkins Medicine. He received his undergraduate and medical training at Brown University, internal medicine residency at Dartmouth, and doctoral training in epidemiology and biostatistics at Harvard. He is board certified in internal medicine and clinical informatics, and an elected fellow of the American College of Physicians, the American College of Epidemiology, HL7, the American Medical Informatics Association, and the American College of Medical Informatics, as well as a founding fellow of the International Academy of Health Sciences Informatics. He was president of ACMI through 2018. His career has focused on how we can represent clinical information to support analyses and inferencing, including comparable effectiveness analysis, decision support, best evidence discovery, and translational research. He has had a deep interest in semantic consistency, harmonized information models, and ontology. His current research focuses on translating basic science information to clinical practice and how we can classify dysfunctional phenotypes. He became founding chair of biomedical informatics at Mayo Clinic in 1988, retiring from Mayo in 2014, where he remains an emeritus professor of biomedical informatics. He is presently PI on a spectrum of high-profile informatics grants from NIH spanning translational science. He has been active on many HIT standards efforts and chaired ISO Technology Committee 215 on health informatics and the World Health Organization International Classification of Disease Revision. Please join me in welcoming Dr. Chute to the podium.
Well, you can tell I'm an old guy, if nothing else. So I'm going to give you a stroll today. This isn't a deep talk. This is a sampler of what some of us are doing in this space. But I want to start with a premise. I want to assert, and I don't think this is a particularly speculative notion, that healthcare, whether we like it or not, has become a profoundly information-intensive industry. No matter what we do, there's some handy things we can do, control costs, improve quality, and so on and so forth. Ultimately depends upon information, and ultimately depends upon patient data. Well, that says volumes about how we represent patient data, or for that matter, any other kind of data, if we're going to leverage it for those kinds of uses. You just can't take an X-ray report and put it into a logistic regression. It doesn't fit. You need some thought to this process. Well, this is supposed to be about big data, so I thought I'd give a little history. This is my dear friend Galileo. You'll note throughout this talk that most of my friends were from the 17th and 18th centuries, which says volumes about my social life. But this is his notebook, and you'll notice, if you look carefully, there's a big blob and some small dots, and a big blob and the small dots in different places. That, of course, is Jupiter, and the moons of Jupiter having different conformations suggesting that it was a mini solar system. And Galileo, in a stroke, with a $3 telescope in his backyard, changed our conception of what we think about as science, the universe, our place in the universe. This was big data in the 17th century. Well, today, of course, the Johns Hopkins manages the Sloan Digital Sky Survey, Alex Soleil's work, and a lot of astronomy is done in silico, at scale, by large teams of scientists who assemble large amounts of data. I mean, there's 1.2 billion objects in this particular data set with a lot of depth for each data element. This has become a large-scale process. The $3 telescope is not going to change our place in history or understanding today. Well, another example, this time in physics, not astronomy. You'll recognize Rutherford, he's somewhat younger, early 20th century, and his tabletop experiment. You remember that, you read about it in high school. And the big question of the day around 1905, give or take, was whether the atom had a plum pudding aspect or whether it had a solid core. We didn't know. And so he put alpha waves against a gold foil, you remember that, and some of them bounced back. And he demonstrated that, yep, if you're going to bounce a gamma ray back, then alpha particle, sorry, then it has to have a very dense core. So this was the beginning of quantum physics. This was the beginning of a quantum notion of the universe. Changed the universe. That table, by the way, is still at the University of Manchester and is still radioactive, but nevertheless, it was a small-scale science that changed our view. Well, you'll recognize this, that Higgs boson thing. That's a person standing in the middle of the cyclotron. It's just a little guy there. It's big scale. So what's physics today? 10,000 scientists, 600 institutions, 200 petabyte of data to discover the Higgs boson, and a modest price tag to go along with it. Only 2 Nobel Prizes. It doesn't seem quite fair, but nevertheless, the scale of science, whether we like it or not, has become a team sport, has become dependent on synergistic data sources. And my point is that biology and medicine, again, whether we like it or not, are entering an era of team science, of big science, where the interdependencies of data sets, data resources, experiments, information, to enable, if you will, the next generation of discovery are going to be demanding. Well, my son's an astrophysicist, and he tells me that those Higgs boson people and the cyclotron people, they actually cheat. Because all they have to worry about are 3 dimensions: mass, energy, he tells me they're the same, I trust him on that, direction, and charge. That's about it. That's all she wrote. You keep track of those 3 variables, you've got it. Well, medicine's a tad more complicated than that. Last I checked, and it's been a while since I've seen a patient, I understand that, but last I checked, there are more than 3 variables involved in healthcare, involved in patient management, involved in medicine. And in parallel, this is one of my whimsy slides. I like to show this. I'm well into my 60s. So over my lifetime, Moore's law, you've all heard about that. Roughly 10 to the 13-fold increase in computing power. What spies used to kill each other to get 30 years ago is now under Christmas trees. And as game platforms. So correspondingly, we've got a 10 to the 10-fold increase in transmission rates. If you remember teletypes, you could actually type faster than the computer. That was pretty cool back in the day. Vacuum tubes to terabyte RAMs, paper tape to petabyte drives, and then a computing factor, a clouding factor. So if you add those exponents up, and I understand that's cheating because it's exponential space, you're really multiplying, right? But we'll ignore that. If you add them up, you get roughly 10 to the 60-fold increase in our ability to manage information relative to where we were when I was born, give or take. 10 to the 60. That is an astronomical number. That's really big. And if we have 10 to this, I mean, could you imagine Google in the middle of World War II or GPS? I mean, the world we live in is quantitatively, qualitatively very, very different as a consequence of the information revolution. Well, if we've already asserted that biology and medicine are information intensive, and we have this profound a capacity to manage and manipulate and understand information relative to where we were when I was born, that has to have enormous consequences on the science and analysis of data in biology and medicine moving forward. It's inescapable. Well, this is an outline of one of the grants I'm working on, the Translator grant, and this was cribbed shamelessly and quite deliberately from a National Academy of Sciences report, 2012. I actually took part in it on precision medicine. But you see that we characterize the notion of disease as planes of information space. So in this particular diagram, they have exposures, phenotypes, epigenome, genome. I mean, the layers are kind of arbitrary. But the point is, disease is a multidimensional concept that has attributes and characteristics from many planes or many domains, rather than being, as my introducer said, I chaired the ICD-11 revision for WHO, and that's kind of a very quaint notion of how to think about disease. You write a title down and you get a number, and ICD-11 is not your father's ICD, but nevertheless, the conceptualization that the National Academy was trying to promote is this multidimensional, multiplanal characterization of aspects and attributes, and together, ensemble, they make a condition or they make a specification of what we think about as disease. And the recognition is that the basic science side of that and the clinical side of that both feed into the process. From the basic science side, you're getting omics and proteomics and genomics and the omics zoo that you hear about frequently. And on the clinical side, you're getting increasingly detailed phenotyping, deep phenotyping of patient characteristics, symptoms, signs, manifestations, characterizations, that I think exceed the previous notions of clinical description. And that they create a new way of thinking about disease, that disease is no longer an ICD code. Well, the other reality is that there's a proliferation in the world today, which is a good thing, of multiple databases, public data sets, public databases that have knowledge, that have data, that have observations. And to be a scientist in this modern era and to map, if you will, these planes of characterization to what is known about genomes, what is known about pathways, what is known about proteins, what is known about variations, invokes a computational challenge of integrating public knowledge databases with these planes of observation and modes of characterizing disease. This is the biology side, and of course, I've left your favorite database off, I apologize. And arguably, this is the clinical side. There are different ones in different spaces, but you get the idea. They're supposed to be exemplary, not comprehensive. The key message is that there are lots and lots of public databases, and that if you want to really excel in making new inferences, discoveries, and associations in biology and medicine today, it's critical that you integrate those sources into the process. Well, here's an example. Animal models. You've all heard of animal models. And if we think about science, if we define experimental science as the ability to create an experiment, to vary parameters around it, and reproduce it an arbitrary number of times until we have experimental evidence that something is so, that's hard to do with people. It's even hard to do it observationally, because the dirty secret about people and rare disease and observation is that we're dealing with a small numbers problem. How many people have a particular rare condition? How many people do you have access to that have a particular rare condition? We're talking typically on the order of scores, maybe hundreds. That doesn't quite bump the big data machinery paradigm. And to enable inferences and collections and experimentation and information that involves large numbers of these observations, we really have to have animal models that correspond to the biology questions and issues that we're asking. So here's just some simple example of eye variations and cornea variations in human, mouse, zebrafish, and Drosophila. We can learn things about these molecular events and their pathways from animal models. We can experiment on animal models. And the goal is to generalize that information forward to human conditions and questions. Well, now not only are we dealing with large public databases of human information, but we have to think about how do we transfold that out into the process and science of experimental observations on animal models that can be integrated with the clinical and translational questions. A non-trivial task. And part of that is because each of these animal model communities, zebra, fly, a worm, Drosophila, I mean, we could go on, mouse, has evolved in kind of a scientific silo over the past 50 or 60 years, in some cases 100 years. And they've come up with their own vocabularies, they've come up with their own terminologies. I'll let you in on a secret. The core focus of my career has been incredibly dorky. My mother was bitterly disappointed. I focus on vocabulary. I focus on how we name things. And the reason I care about how we name things is because when we try to integrate data across different sources, public databases, animal model communities, and the like, guess what? They've named the same darn thing differently in those communities. So we're left scratching our head, well, does this really correspond to that? Well, we could look at the underlying biology and say, yeah, it does. We could look at the underlying biochemistry and say, yeah, it does. And eventually we're doing that. But it raises the challenge of how do we do large-scale data integration and cross-translational research across these myriad of public databases, absent a way of synthesizing and connecting like with like. It's the ontology problem. And this is what I've characterized as, in this case, I have the biology community and the medicine community. I mean, we could put any number of poles on this axis. And I've just got some characterizations. And there's no data on this slide. I made it up on the back of a napkin. But it illustrates a point in that there's a lot of activity on the basic science side, which is orange, and the medicine side, which is green. I can do that because I'm Irish. And what I use this slide to illustrate is what I characterize as the chasm of semantic despair. Meaning these communities, historically, have come up with their own vocabularies, their own ontological frameworks for describing their observations and their work. And, the basic scientist and a clinician at a cocktail party, it's a very interesting conversation. And it's not as often scientific as you might think. Partly because they work in such different communities. Another example of this is our colleagues from machine learning. The machine learning community and the biostatistics community. I mean, for years they would have catfights over who was smarter and who had better machine reason tools, until they discovered one time that layered neural networks and stratified logistic regression were mathematically identical. They didn't know that for a decade, because they used different terms to describe the underlying principles and observations and mechanisms. And yet they were doing the same darn thing. They had come to the same conclusions. And we have that in spades across science. Well, ontologies will save the day, the ultimate converter. I won't bore you on what I think an ontology is. I can and often do go on for days on what an ontology is. But simplistically, it's a way of organizing knowledge. And more pertinently, in a modern information science era, it's a way of organizing knowledge that allows connection with logical components, specifically description logics. Description logics are a computable subset of first-order predicate logic, that stuff that Aristotle and those 13th century monks would work on, syllogisms. But as a practical matter, first-order predicate logic is not computable. If you take it down a level and make a subset of first-order predicate logic, you come up with description logics that can create ontologies so that you can define a body of characterization that is computable, against which you can make inferences, and allows cross-linkages between and among ontologies in the community. The other reality I want to point out is that if we think about where knowledge comes from, and this is my trademark slide. I've been showing this puppy for 30 years. But if we think about it, Information ultimately begins with a patient in biology and medicine, and that pertains to basic science as well, if we're talking about biospecimens. You create data, you make inferences, you generate medical knowledge, you get tenure, you're done, you go home, right? No, well, with that medical knowledge, if you're really good, you reinsert it into the system, knowledge management, decision support, and you make this virtuous cycle, this samsara, this wheel of learning, that can inform future processes by observation and inference. What keeps that circle growing around? Well, you know me, I'm a one-track guy. To the extent that medical knowledge and patient data are sharing terminologies and data models, that process can happen a lot more quickly. That's not what happens in the real world. The decision support people have their own quality assurance mechanisms and so on forth. The clinical people have different, it's not really aligned. And it's the importance of having the framework on which big science can be conducted consistently represented in whether you invoke the dreaded ontology word or not. As long as they're calling things the same in the communities, that's the bottom line. Well, long history to this. This is John Grant's observations in the bills of mortality. I was a little bummed. When I was at Mayo Clinic, we had the 1665 edition, and that was the third edition, and I thought that wasn't cool. You want the first edition, right? But it turns out this third edition had neatsy cool foldouts at the back that the others didn't. And these foldouts, I mean, this was a hugely influential piece of work. Because what happened was this was the first time that diseases were systematically counted in a population, causes of death actually, and that the statistics were generated year by year on totaling these causes of death and allowed them to make interesting inferences like, gee, some diseases are epidemic, they occur in flurries and plumes and then go away. And other diseases are endemic. They hadn't quite grasped that. I mean, they understood the concept of an epidemic, but they didn't know, structurally how these things worked. These were generated, of course, so that the king didn't have to go where the Black Death was. But in general, and there's some fascinating diseases on this. This is my favorite, found dead in the streets. I'm not quite sure what the underlying pathophysiology, never mind the genomics, of being found dead in the streets really is. So it wasn't a perfect classification, but you get the idea that there was becoming a systemization of naming things. I can't emphasize that point strongly enough. Some of the names are dorky, but of naming things so that you can quantify it and count it. And this was a survey we did last year on the use of ontology in the literature. Here's our friend ICD with all its warts and flaws, and other ontologies that are not quite as broadly used in either the clinical or the basic science literature of biology in any event. And for your evening reading, the Census of Disease Ontologies, which we published last year, kind of reviews what ontologies are relevant to big science, to data science, to biological discovery and translational research as we move this forward. Well, remember that multi-plane characterization of disease that we had before. When we talk about an individual, we have phenotypes, we have genes, we have environment. What makes them different? What makes us different? What results in some people having one natural history and other people having a very different natural history with respect to disease? And if we think about it, a disease can be described as a collection of phenotypes, observations, characterizations, so that we have, if you will, the characteristics of a patient at the top and the characteristics of a disease at the bottom. And you can see where we're going with this. As a way of creating diagnostic aids, to the extent that you have simplistic mapping of patterns or profiles of what constitutes a published disease, and you can generate a profile of what's going on with a patient. I mean, this is not new. DXplain, I think, was experimenting with this 40 years ago, and other software. But the principle is that you have a concept of mapping a profile and characterization of a disease with a profile and characterization of a patient. So if we think of a patient with various characteristics drawn from different axes, if you will, or planes, and models of different diseases, we can quickly see that one fits more closely than another, and literally map associations and diagnostic findings in a way that, this is simplistic, because some of these variables are continuous, some of these variables are multifactorial, some of these variables are binary. That makes it easy. So it's not quite matching up the dots. But the principle of this kind of pattern recognition and matching, which in turn is dependent upon having comparable and consistent naming of these underlying patterns, is what we're all about. So computational comparison of human models and phenotypes with human disease. Again, if you have connections between and among human phenotypes and disease animal model phenotypes, you can start to make inferences and computational associations from these connections. And my colleague Peter Robinson, of course, demonstrated that fairly convincingly some time ago. Well, you'll recognize this notion of public data and patient data going into, if you will, an inferencing funnel. And the biomedical translator, which is one of the grants that I'm PI on from NCATS, the same people that bring us the CTSA, has funded an effort where we generate data in these two spheres, render it in a common framework, append it or annotate it with ontologies, with ontological linkages, that allows us to take the rendering of the underlying disease and the knowledge sources and to computationally make associations. So the aspiration, and I won't say it's a reality because we've only demonstrated it on, frankly, a handful of ground truth cases. We haven't discovered anything yet. But if you have the manifestation of a patient that is caused by a physiologic pathway, that is associated with a metabolic pathway, that is associated with a protein conformation and function, or misfunction as the case may be, that is associated with a protein sequence, that is associated with a genetic translation, either post-translational or pre-translational, that is associated with a variation, a variant, you can make the inference all the way from manifestation to cause in a similar kind of way, computationally. Correspondingly, if you know what those manifestations are, and then take that further in a speculative way and say, all right, we have these proteins, we know these proteins are involved in that pathway. Are there other protein targets in that pathway that we can influence, perturb with drugs, with other kinds of interventions, and have a reasoned association of, to stop that manifestation, chasing down several layers of public data sets, if we have this pathway with these proteins, these known drugs that can either accelerate or inhibit or bypass or otherwise perturb that system, can we create a putative question of, does that drug that is computationally predicted to interrupt that pathway have a chance of benefiting that patient? Now, we all know that the potential for harm, the potential for perturbing unintended pathways is huge. It doesn't mean just because you discover something that it is going to be something you would try in a patient, but it is certainly practical in the context of hypothesis generation and coming up with explorations and potential therapies and clinical trials that might not have occurred to a human being simply because the magnitude of the data in both the public data sets, the pathways, the databases, the proteins, the perturbations, the drugs, exceeds the capacity of a human mind to possibly integrate. And the goal of Translator is to facilitate that integration computationally. So it's done by taking our layers of expression and pathway. You notice we've changed it a little bit, gene variants, models of disease, function interactions, treatments, microbiome, and so on. We graph inference using description logics and ontologies across those layers, and we can ask questions of those layers speculatively moving forward. This slide is self-evident. I shouldn't have to say anything about it. It's not really that complicated. What it does is we've got genomics, model organisms, humans in one category. We have sources of data, genes, variations, ontologies. We have names for those data sources. We have ontologies with which they are annotated, so you're seeing some interweaving to common ontologies. And then the knowledge graphs that I just described operate across those interwoven renderings in an environment. We've called it BioLink. It's an open source tool, open source environment to enable questioning and leveraging and integration of sources of that level of complexity. We described this a little more generally. I think last year we published our review in New England Journal on classification ontology and precision medicine that we think makes the case. I mean, it's not often you get an article on such a technically arcane topic in a clinical journal, but they thought it was relevant and at least ready for prime time to think about. I'm going to change gears a little bit to show or to describe another effort that I'm a PI in, and that's the Center for Disease to Health. And this very Friday, that's 2 days from now, we'll be presenting our efforts about the Center for Disease to Health, which is really a coordinating center across the CTSA informatics cores to integrate how the informatics cores can really operate as a network. A lot of the themes I was just babbling about, common naming, common models, common frameworks, computational interoperability, pertain when we think about the CTSA or the network of CTSAs. You remember what you call the ICTR to the rest of the world is a CTSA. Hopkins does the same thing. Is what we're presenting. And I'm not going to give you that whole presentation, just some slides to say that one of the elements that we're focusing on, and many of the folks in IT would be deeply familiar with this by now, is FHIR, or the Fast Healthcare Interoperability Resources. We're working very closely with HL7. We're pursuing creating a new translational research working group at HL7. And Graham Grieve, the developer of FHIR, has characterized it as the web for biology and medicine. Remember, I've been focusing on ontology. I've been focusing on names. Well, the reality is a pile of names isn't sufficient. They have to have context. They have to fit into a model. They have to fit into some kind of framework or slot so that you can do something intelligent with those names, that they have relationships to other names. And that's effectively what FHIR is. So the bad old way was every CTSA being a pillar unto itself with different databases and data sets and point-to-point connections and so on and so forth. And what we're envisioning is the FHIR-free way where we can generate data derivatives, spin-offs. Obviously, we're focusing on large-scale data models like Odyssey, OMOP, and PCORNet, but the reality is we could also equivalently generate protocol-specific data sets and projections from a common frame of clinical data. And the way that helps us is think of FHIR as Lego pieces. We have observations, we have reports, we have medication administrations. Each Lego piece has characteristics. Here's the observation Lego piece. So we can take one Lego piece off the wall and create a PSA observation out of it. We can take another Lego piece observation, create a serum sodium, and these things are interoperable. These things are self-describing objects that allow computational tractability between and among them, putting clinical data in a very different reference frame than it's been historically. And as far as its use in translational research, discoverable data, building cohorts, more information from the data, interoperability with other hubs, making the CTSA function as a true network across the community. And yes, we're a bunch of crazy kids, that's certifiable, but it's also true that NIH is persuaded that there's something to this. And there was a notice that came out just a few months ago from Francis Collins. It was actually a White House announcement Don't get me started on presidential politics. But nevertheless, to encourage researchers to use FHIR for interoperability in clinical research and translation. The real elegance is that FHIR comes out of the clinical community. They're paying for this. They're generating it. And trust me, the resources of the clinical community are orders of magnitude larger than the resources of the research community. So if the research community can leverage what the clinical community is generating in terms of data interoperability, the Office of the National Coordinator, everybody who's CMS, they're all in love with FHIR. I'll close briefly on just describing what we're doing back home and what hopefully, I was speaking with Professor Davis, what hopefully we can engage the University of Maryland to partner with us. And that could be the stalking horse for a CTSA-wide framework and engagement for translational science, is what we call our precision medicine analytics platform. Simplistically, it's a way of integrating data into a common framework from multiple sources, enabling discovery, and then closing the loop by putting that discovery back into clinical practice. It's pointless to have just discovery if we can't instantiate it back into a clinical practice world. We started it with for those of you who are Apache fans, we started it with a standard suite of what I call next generation database, data analysis, data management tools. You all know that Google has changed the face of the planet, the way it manages vast amounts of data. That's done on a software platform that is vaguely characterized as HBase and Hadoop that allows basically there's no practical limit to the scale of data that you can incorporate into this. This is very different from the way we thought about relational databases, fundamentally different. So we're leveraging those kinds of environments in partnership with Microsoft and Azure to take in data from various sources, Epic, REDCap, patient-reported data, home monitoring devices, whatever might be relevant, into a data commons with a common research environment and a common analytic environment. Just a word on the analytic environment. We're using Jupyter, which for those of you that are familiar with it, it's a fairly standard data science tool, widely used through the data science community. It allows you to create a clear record of what analysis has been done, what manipulations have been done on the data, what transformations have been done, and replay them quite easily so that you can create, and has a good client. It can handle text and math, code results. It handles Python, it handles R, it handles the standard tools. The data that we're also bringing in is not just the electronic health record data, but data ideally from the source. We can go to laboratory systems and get more rich metadata, imaging systems and get more rich metadata, REDCap, of course, in terms of patient-collected information and the like. And then manage that in a platform for analysis where the data is integrated in a common framework in a cloud. I mean, Microsoft Azure is a cloud. So the capacity to expand dynamically for analytic, for large-scale analysis is trivial. That's what clouds are for. And it is becoming a very scalable, very exciting framework for us to think about information. So I've prattled long enough. My summary is that biomedical practice and research are data, information, and knowledge intensive. That is a truism, I would assert. Comparable and consistent data is prerequisite. Clinical data is broad, deep, and complex. Inferencing on clinical data is not the same as Google, and that ontological frameworks are really crucial to help us scalably integrate this information in an interpretable, computational way. And with that, I probably have a minute or two for questions. Is that right?
Thank you, Dr. Chute. Wonderful and very entertaining presentation. Thank you. Questions, comments?
Does the data in the PNED, does that, I mean, it's coming from Epic, so there must be some identifiers there. Does it get de-identified in some places?
Yeah, that's a good question. In the Hopkins precision medicine analytic platforms, the data in its commons format and framework is fully identified. And when we speculate about making a Maryland-wide data set or a Mid-Atlantic data set, we might have those identifiers managed by the CRISP organization, who already has a large data. But, you know, Hopkins wouldn't know Maryland identifiers and vice versa, but we'd know that, you know, there's a common framework here. When we deploy data or disclose data to an investigator, unless there's a need for the investigator to retain identifiers, we de-identify it at that time. But we manage it internally in a secure cloud as with identifying data.
Who pays for integrating all this data and also for accessing these data?
Yeah, the question was who pays. We have, for the moment, a grant from President Daniels of Johns Hopkins in partnership with the Applied Physics Lab to build the underlying data framework, data structures, data integration environments. So in that sense, the university is subsidizing its development and the integration of that data. The provisioning of the data is something historically, and I know Maryland has a different model, we've charged investigators for. If they want to get clinical data, we charge them simply because the dean's office does not provide, I actually run that operation under my space, and I get lots of angry email from investigators who don't understand why they have to pay for it. And the reason why they have to pay for it is because our university has chosen not to subsidize that phase of research. So initial data query and integration is an hourly charge of analysts that do it on their behalf. We are thinking for the precision medicine analytics platform of various financial models because the institution is not going to subsidize it forever. We want it to be a cost-neutral environment. But the argument is that, you know, if we make this really good, the actual cost to the investigator will be quite modest to do what they really wanted to do, because it can be done so much more quickly and efficiently than it would otherwise be able to be done. They certainly couldn't do it themselves any cheaper. That's the main point.
Chad? Quick question. You know, in terms of big data science and genomics, traditionally it's academic medical centers like Harvard and Mayo and Vanderbilt that's been leading the charge. But more recently, it's been sort of non-academic places like Geisinger, Inova, and Intermountain. They don't even have a medical school. What's in it for them, and what's the end game for these people? And are we missing out by not participating in that kind of data science effort?
Yeah, I think Hopkins concluded we have the potential to miss out, which is why we're investing and potentially partnering. Geisinger just founded a medical school, by the way, so they're turning. And Intermountain for many, many years was joined at the hip with Utah. They had a divorce about 2 to 3 years ago. But to answer your question, I think people aspire to have big data, omics data, help them inform specific practices that would subdivide otherwise heterogeneous populations and diseases. I mean, if you look at asthma, I'm not an asthma expert by any means, but people tell me, people who know, that you can, even without omics, you can peg asthma patients into steroid responders, beta agonist responders, and so on and so forth. And it's and there are increasingly evidence that if you can tailor a treatment to a specific patient, the Vioxx quagmire. My wife loved Vioxx. I mean, she swore by it, but it's off the market. And it turns out there's probably only a small subset, you've heard this before, right? There's only a small subset of patients who probably have adverse reactions to an agent like that, where the rest of people might benefit enormously. The vision is, to the extent that you can distinguish subtypes of patients within a disease population, they're going to have preferred response to modes of therapies, not just drugs, but to all kinds of modes of therapy. Delivery can be delivered more quickly, accurately, and effectively, and you avoid potentially years of morbidity with inappropriate treatment or ineffective treatment. That's the vision. That's what they're seeking. Is the reality there? I would say yes in the world of cancer. I mean, we already know that we can characterize cancer and tumors, whether they're going to be responsive to a protein kinase inhibitor or not, whether they're going to be responsive to other modes of therapy or not. So we're starting to see the beginnings of it in a practical way, but I acknowledge that we have a ways to go. This gentleman's had his hand up for a long time.
At the risk of sounding like a dullard, you've used the word ontology and ontological frameworks throughout your talk. If I remember my Latin correctly, onto comes from the Latin to be.
Yeah.
Ology is the study of. Ontology is the study of existence or being.
Yes.
What is an ontological framework?
Yeah, fair question. And I confess that the computer science community has co-opted the word about 30 years ago, most ungraciously. I mean, if you think of a Platonic a universe outside our own, that is what exists, that is ontology. And the notion that you can have a plurality of ontologies is an oxymoron in that philosophical sense. I accept that. This is well known. When the computer science community started assigning the word ontology to an enumeration of aircraft parts for the Boeing 777, which they did, when you describe what a Boeing 777 is in terms of its components and how they fit together and the relationships among them, they've used the word ontology within, if you will, a closed world domain, in this case a Boeing 777. Correspondingly, we're using it in disease states. We're using it in domain states like genomics, proteomics, and the like. So the word has been co-opted, perhaps badly, to describe a universe or a space that is at least conceptually self-contained. And what we're striving to do in translational research is to take these self-contained worlds that are described in ontological frameworks, be they proteins, be they genomics, be they biological pathways, be they drug catalogs and pharmaceuticals, and navigate them to make inferences and connections across them.
Carl?
Yes, are you concerned that clinical data is not clean enough
Yeah.
to avoid this classification bias and power to make inferences?
Yeah, excellent question. Yes, is the short answer. I'm deeply concerned that clinical data is a mess. It's full of missing values. It's full of misdiagnoses. It's full of error. It's full of misclassification. Yes, is the answer to that question. So you have to ask the counter question. Would we be better off with our own intuition and judgment? I interned in an era of what I call the expert anecdote, where the attending knew all, no offense. It was kind of this priestly learning at the feet of your seniors in a way that, the chief resident, or the intern grew up to be the chief resident, grew up to be the chief of medicine, and he did pretty much what he was taught to him by his chief resident. So there was this oral tradition, this folklore. That's the history, to a significant degree, of clinical medicine. To the extent that we can transcend that and bring more and more evidence to bear, is it perfect evidence? Clearly not. And the theory is, and this is the whole premise of big data, if you've got 5 observations and 2 of them are badly biased and flawed, you're not going to come to any kind of useful conclusion. If you have 5 billion observations and 2 billion of them are biased and flawed, you can actually get some signal out of those 3 billion. And the idea is, if we systematically examine data, and then the other adage, of course, is what gets used improves. To the extent that we're actually using data to make inferences, and people start to understand that, then they're more invested in the data quality than, frankly, they are today, where a major use for it is to create a billing code, which for many academic physicians is some abstract thing that happens in an office down the hall.
Jeff, who owns the big data? If you're getting the connections, and that connection is specific to the data that was used to generate it, and does that differ depending on whether you're in the United States or in Europe?
Yeah, so who owns the data? Excellent question. For many of the public databases, Melissa Handel, my colleague, she's at NLM today, actually, talking about data licensing and data ownership issues. A lot of, the Creative Commons licenses, the CC0, the CC BYs, and so on and so forth, make very clear what the uses of the data might be. It turns out most of the biology community has been committed to open public data for a very long time. This was a tradition that was started in the early sequencing days, where Francis Collins would not fund the sequencing centers unless they made their data public almost immediately. So it was sort of artificially instilled, and I think the basic science community has continued to a significant degree in that line. Clinical data is another matter entirely. And it's not just because of data ownership. It has to do with privacy and confidentiality and other kinds of things. That's where we dip our toes into, I could give 12 more lectures on the notion of federated analytics, where you have an analysis within a clinical environment behind the firewall. And historically, you'd create a point estimate, and you'd pool the point estimates and maybe the variances and come up with a conjoint point, kind of a meta-analysis. Not very exciting. What we're thinking and doing and experimenting with is creating not just a point estimate, but partially performing the analysis, be it a machine learning analysis or a statistical inference, behind the firewall. And instead of creating the final parameter estimate, you create tables of covariate tabulations. You create a matrix of information that's de-identified because it's just a matrix of tabular partial results. And then you pool centrally the matrices that arise from behind the firewall and get a much more robust analysis that almost approximates, in terms of reliability, consistency, bias, and everything else, what it would be like if you could analyze the row-level data itself. So we're seeing more and more truly federated analytics that have this almost the same statistical power as a full row-level analysis. I don't know if that answered your question, but
Well, Jeff reads a lot of George Orwell.
Great presentation. I was thinking about your graphic where you had your different layers and the planes of characterizations, and just wondering about your thoughts on how you socialize this to other stakeholders beyond researchers. Patients, well, there was a question earlier about licensing and data ownership. They're historically skittish about how information gets used and linked or delinked and inferred and interpreted and then subsequently shared. We have data vendors now who are nervous about linking 5 digits of codes instead of 3 digits. And so that's sort of the level where we are. And then I look at a graphic like that and I think, well, how do we get there in terms of that socialization beyond research?
yeah, no, that's an excellent question. I think it's an important one too. I could be glib and say, well, we've got the 18 safe harbor identifiers and 3-digit zip code is okay, 5-digit is not under that space, and we adhere to safe harbor and all is good. We all know that's not true. We all know that there's a risk of re-identification with any distributed data set. I mean, if the one that always catches my attention is Brad Malin would say, if you give him 5 hemoglobins in sequence, he could tell you who that is. Because it turns out a sequence of 5 numbers to 3 decimal places is fairly fingerprinty. So there are ways of re-identification, and re-identification risk is a legitimate concern because of data integration and inferences that can be deleterious to the people and the communities involved. That's not lost on most of us. I think the finesse, at least for the time being, is what I was babbling about federated analytics before, that to the extent practical, sensitive information, patient-level information, is analyzed in as rigorous and as safe an environment as we can achieve. For Hopkins, it's our precision medicine analytic environment. We ain't going to let Maryland see our data, but we might let Maryland see our matrices of sub-results that are arguably completely de-identifiable, or that the re-identification risk is arguably zero. So I think you raise a good point, and it's something that, in fact, Jeff Kahn, who leads the Berman Bioethics Institute at Hopkins, is meeting with members of the research subcouncil of the Data Trust. I co-chair that at Hopkins with Stuart Ray about this very question. How can we ensure our obligation to our patients to preserve their privacy and confidentiality, while at the same time fulfilling our social obligation of discovery and dissemination of learning.
You should last question.
Thank you so much for your talk. I was intrigued by the fire highway. So I think that as we know that the fire is from clinical side,
Right.
but we also know that the adoption of fire in hospitals. So I'm wondering, some hospitals like Duke or Utah, I think they implementing fire, but not many hospitals have capability of implementing it. So what do you think, what is your point about the fire adoption in hospitals among those CTSA organizations?
Fortunately, the Office of the National Coordinator and regulatory requirements are basically putting the onus for implementing fire interfaces on EHR vendors, not on hospitals. So that if you have Epic, or if you have Cerner, or if you have one of the major systems, you have fire capability. You may not know how to use it, and you may not deploy it, but it's there. It's just a matter of turning it on and using it. And I don't minimize that step. It's a big step because it's conceptually very different from the way health IT organizations have thought about data. I mean, it's a very modular data science way of an object frame for thinking about clinical data. HIT communities don't historically think that way, but the capacity is there. It becomes more a socialization and a culturation and an education of the community to leverage that capability that's technically present. Okay. I can't shake your hand.
Thank you so much.
Yes.
Especially when the really said overruled you with some delicious lunch.
We'll wait up here.